Consideration of biological potentials such as resting potential and action potential. In the lecture part 1, we begin from the resting potential. Starting from this lecture, we begin a new section of physiology, electrical signaling and excitable tissue. The main excitable tissue are nervous and muscle tissue. Any cell of the body is able to respond to stimulation by some non-specific changes in its functioning, but the cells of the excitable tissue respond to stimulation by a specific reaction named excitation. For neurons, this excitation process appears as generation of an impulse or action potential, which then is sent down the axons to the other excitable cells, for example, neurons. For muscles, excitation results in their contraction. First, impulse comes from the neuron through the axon, then through specific structure called synapse, impulse is conducted to the membrane of the muscle cell, and finally, muscle cell membrane generates its own action potential, and that makes it to contract. These biological potentials are electrical signals that used for the fast transfer of information by impulses between neurons, and also impulses can be sent from neurons to the other excitable cells, mostly muscles, but also some endocrine cells may respond to impulses by their secretion. It should be mentioned that the electrical signaling is much faster as compared to the chemical signaling, and also is much more precise as impulses are sent directly to the target cells. So it's a type of communication between cells that developed later in evolution, and this is faster. Before we consider the mechanism of formation of these impulses, or action potentials, it's necessary to understand the basis for all potentials generated by excitable cells. And this is the basis, which is resting membrane potential, as membrane of the excitable cells is constantly charged at rest and has the specific level of potential. Resting membrane potential is a potential difference that exists across the cell membrane in a resting state. More exactly, we can say that this potential difference is difference between the charges or potentials of the inner and outer sides of the membrane, or simply we can say between the cytoplasm interior of the cell and the interstitial fluid that surrounds the cells. And as we can see, the surface of the cells, the outer side of the membrane, is charged positively, while the inner side of the membrane is charged negatively. And exactly this negative charge of the inner side of the membrane is considered to be the resting membrane potential. And potassium ions shown inside the cell indicate the main reason of resting potential formation, while potassium permeability for the cell is present, while sodium permeability at rest is very low. Let's consider this in detail. Let's recall first the cell membrane structure. This is bilayer of phospholipids, basically. And also there are some proteins present on the inner or outer side of the membrane, or some proteins are integral ones, which pass through the whole membrane. Phospholipids is the main portion of the membrane, main structure. Then some proteins, including the integral proteins. And as you can see, between phospholipids, hydrophilic substances cannot pass simply. And ions are classical hydrophilic substances because they are charged particles, and they dissolved in water well, because water makes H plus and OH minus ions, while in non-charged phospholipids, these substances cannot simply pass. Therefore, for opportunity for ions to pass through the membrane, there should be specific or non-specific channels. Ion channels of the membrane first exactly are divided into the two fundamentally different groups. Non-specific channels which do not have gates. No gates, therefore they are open constantly. Also, they are called leakage channels because at first they were described exactly for the potassium which leaks from the cell through these channels, goes out from the cell. And there are specific channels which have gates, therefore they can be present in their closed state, and under certain conditions they become open. Then, let's consider first non-specific ion channels. These are simply the pores between the membrane phospholipids. It's one type of opinion of these channels. And look, when phospholipids of membrane that are constantly in little movement, slight movement, they form a little bit larger pore between phospholipid heads, and so during this moment ions, specifically potassium, can pass through these channels. To make some ions to pass through the channels, it's necessary to have the force, moving force for their movement, and this is the difference of concentration. If amount of ions is higher, concentration is higher at one side than the other, so ions predominantly will move down the concentration difference. These channels are not existing constantly, and during the next movement the heads of phospholipids will become again closer to each other. But similar channels can be formed in other areas, and statistically their number per time unit is approximately equal. So they appear in one area, disappear and reappear in some others, thus providing for the ions opportunity to move down the gradient. And so permeability, therefore, is considered constant because of practically constant number of channels per each moment and per each square unit of the membrane. And the resting potential is formed exactly due to these channels. Recent investigations offered the other possible structure of the non-specific potassium channels. The channels are formed by the integral protein loops that selectively allow potassium ions to lose their hydrating spheres and pass through the membrane. But these channels were identified in some bacteria only by the moment, so we cannot say that such channels are present in humans as well. Anyway, regardless of exact structure of these channels, some properties are stated exactly. First, they are not gated, so they are open constantly. And the second, they are much more permeable for potassium ions, regardless of the exact mechanism of their selectivity. They are much more permeable, and difference in permeability, according to classical counting, is considered to be 25 times, and some sources suggest that even 100 times higher permeability exists for potassium as compared to sodium permeability. So, in contrast, the specific ion channels are formed by proteins, and in turn they are divided into two groups, depending on the mechanism of their opening of their gates. One group is voltage-gated channels, which become open under certain change of the membrane's potential. In most cases, this is depolarization, decrease of negative charge of the inner side of the membrane. And also there are ligand-gated channels, which we already considered in chemical signaling. They become open when certain ligand binds to the protein that forms the channel. In both cases, change of the potential or binding of some substance to the channel proteins results in change of conformation of proteins. This change can be caused by redistribution of charges between parts of molecules. Molecular forces changes between parts of the molecules, so that the portion that closes the channel, the gates, become removed somehow, and channel become passable tube that allows the ions to move. Voltage-gated channels exactly are responsible for the action potential formation. But let's come back to the resting potential. Resting potential is based on a passive diffusion of ions through the non-specific or leakage channels. Open at rest. They are always open. And driving force for ion diffusion through these open channels is the ion concentration difference. So the ion diffusion is directly proportional to the ion concentration difference. D stands for diffusion and delta C is the difference of concentration. So you may understand the greater is the difference of concentration between two sides of the membrane, the greater diffusion is therefore. Let's observe this ion concentrations difference, most important for the membrane resting potential. Two main ions involved into electrical processes. Number one are potassium and sodium. And inside the cells, their concentration greatly differs from the concentration outside the cell. Potassium is intracellular ion, and its concentration outside the cell in the extracellular fluid is much less. It's usually between 4 and 5, and let's say simply 4. Then, for sodium, which is classical extracellular ion, concentration inside of the cell is much less, and outside concentration is around 140, 145, and so on. So you see, for both ions, there is a difference of concentration. For potassium, this difference is directed outside, while for sodium, this concentration difference is directed to inside. And now it's important to understand the selective permeability of the membrane, because not both ions can move similarly. The concentration gradients exist for both ions, but permeability is not equal. This is selective permeability. So it's not equal, and some ions selectively can pass better, and these ions are exactly potassium ions. Classical ratio of permeabilities is considered to be 1 to 0.04 to 400ths. We take potassium permeability as for 1, and you see it's 25 times less the sodium permeability. And as I already said, in some sources they suggest that permeability difference is even up to 100 times. But anyway, potassium can easily go out of the cell, while sodium, much less easy, can go inside of the cell down its concentration gradient. And practically these two basic reasons, difference of concentration and difference of permeability, create resting membrane potential. But now it's important to understand why the resting membrane potential is constant. If we measure the resting membrane potential using specific techniques, it shows a perfectly straight line without any oscillations around the constant level. How it can be achieved? Let's recall, there is a difference of concentration, and also there is opportunity for potassium to move out of the cell down the concentration gradient. But these concentrations given in the table are given for the living cells. And these living cells nevertheless keep the concentration difference still great, 140 and 4, regardless of the ability of potassium to go out. How it can happen? Why potassium does not become, like, let's say, 72 and 72 in both sides, with full balancing of the two concentration difference? Because there is opportunity for potassium to move. And it's necessary to understand that constancy of the resting membrane potential is maintained due to the dynamic equilibrium between the two oppositely directed forces. First force we already know. This is the concentration gradient that makes potassium ions to go out of the cell. But there appears immediately the electrical driving force directed back to the cell, because inside of the cell, loss of potassium results in predomination of the negative charge. And in reality, this negative charge attracts potassium ions back with equal force, equal to the concentration gradient. Therefore, these two forces are balanced, and really potassium can move through the channels, but it moves in the same amount out and the same amount back. So the potential is kept constant. And let's observe this mechanism of formation of the resting membrane potential on the scheme of the model. let's assume that this is the nervous cell. Of course, its shape is not square, but it doesn't matter. And also you should assume that we start from the moment when potential is non-existent at the moment. In real situation, potential exists always, and we cannot observe the moment of its formation. It's present and kept. But let's say that initially inside of the cell, charge is absent, and potassium concentration is high, while outside, and also the potassium ions with their positive charge is fully balanced with anions existing, especially the anions that located nearby the inner side of the membrane. And these are large anions, predominantly, for example, these are COO minus groups of the amino acids in the total structure of the proteins that are located nearby the inner side of the membrane. So these large anions cannot move out of the cell, they remain there. And then let's see, there is a difference of concentration, and there are the non-specific channels that allow potassium to go out. And potassium starts to move out down its difference of concentrations, proportionally to the difference of concentration. And so each ion that leaves the cell leaves one uncompensated negative charge inside. And positive charges appear to be outside. The more potassium go out, the more gradually negativity inside becomes. And therefore, finally, we establish the negative charge that becomes strong enough to bring potassium back. And the force, electrical force, at certain moment becomes equal to the difference of concentration. And then dynamic equilibrium is established when two forces are balanced. And now ions can easily move out, but with the same ease that they can go back because of the negativity attracting the potassium ions. And anions cannot move out of the cell, they cannot follow potassium, so they attract back the small potassium ions. And so we have this balance when dynamic equilibrium is established and resting potential is constant. Using the existing values of the concentration difference and permeability, we can even calculate the value of potential which brings this dynamic equilibrium using the so-called Nernst equation. But the Nernst equation, if it doesn't take into account the sodium permeability, shows the values which are more negative than real potential is. And here we should take into account that potassium permeability is much greater than sodium, but nevertheless the sodium permeability is not zero. It's very little, but nevertheless potassium, not only potassium goes out, but sodium in a smaller amount may come into the cell. And so potassium ions with their even much less permeability nevertheless gradually start to become accumulated in the cell. And potassium and sodium ions are positively charged, and they decrease this negativity which already achieved in the inner side of the cell. But theoretically it can decrease slowly, but decrease the negativity inside the cell. But we know that it doesn't happen. As I already said, the resting membrane potential, when cell maintains the resting state, this potential kept at a steady level without any oscillations around. So somehow the cell prevents accumulation of sodium ions inside the cell and keeps the resting potential constant. And the structure that helps to maintain this sodium law in the cell and removes the sodium that enters the cell is the sodium-potassium pump. This is a specific molecular device made of proteins inserted into membrane of the cell, and this is sodium-potassium ATPase. This is a pump, but at the same time it uses the ATP energy. Therefore, it splits the ATP, and this part of molecule of this protein is able to split ATP, so it's called ATPase. It's a synonym of the sodium-potassium pump. This molecular device is designed in such a way that at first it has the part of molecule able to split ATP, and also there are 3 binding sites for sodium at the inner side of the cell, and 2 binding sites for potassium ions at the outer surface of the cell. And when all these binding sites become occupied, the ATP molecule is immediately broken down to ADP and phosphate, and energy released from ATP breakdown is used to change the conformation of the protein, so that inner part with the sodium ions becomes transferred outside and potassium ions become moved inside. Notice both transfers occur against the gradient. Against sodium is high outside, but nevertheless we move sodium outside. Potassium is high inside, and we move some more potassium inside. Both transfers against gradient, so it's classical active transport with energy use. And due to constant work of this sodium-potassium pump, we have constancy of the resting membrane potential. But you should understand that it's not the main reason of the formation. It's just mechanism that prevents accumulation of sodium. And if constantly potassium that enters and that goes out of cell can be returned back only by pump, it will be not possible to maintain the resting membrane potential. And this is just an additional reason for maintaining. And main reason is based on the 2 things: difference of concentration and difference in permeability. So the potassium permeability only is high. And therefore, all attempts to make all escape of additional potassium is passively, immediately compensated by returning of potassium back. Just imagine if accidentally more potassium leaves the cell per time moment. It creates greater negativity inside, and this greater negativity automatically makes more potassium come back. Therefore, there is no deviations of potential. And at any moment, if potassium movement outside becomes a little bit higher, accidentally, then the restoration occurs automatically, immediately, and this does not allow the potential to be changed. Just sodium that enters the cell gradually results in, activates the pumping, and then sodium excess is removed. At rest, there is no much work for the pump. Notice that pump also participates in creation of negativity inside the cell, because the cell loses 3 positive ions and gets back only 2. Therefore, with each cycle of the pump operation, there is 1 more negative charge created inside the cell. So this pump is considered to be electrogenic. There are some pumps that transfer equal number of ions, so it doesn't create the negativity additionally. But pumps in the neurons and other excitable cells membrane usually are electrogenic, and they are additional reason that creates negativity inside the cell. So, as a result, we can observe the charges distribution between inside and outside of the cell. Generally, inside the cells, we have not only negative charges, there are positive ions as well. Correspondingly, the extracellular fluid contains not only positive ions, but negative as well. But if you observe the surface charges, they all are positive in the membrane. And inner side charges all are negative in the cell. So this difference of charges exists between inner and outer sides of the membrane. Average resting potentials, therefore, are, for neurons, it's between -60 to -70 millivolts. It's a little charge, but it can be measured. For muscle cells, it's more negative. For skeletal muscle cells, it's -90 millivolt. So exactly this is a charge of the inner side of the membranes of corresponding cells. If we need to draw the potential, the resting potential figure, this is simply straight horizontal line. If the vertical axis shows the level of charge, and downward will be negative charge, -40, -60. And this is a line that shows the resting potential of a neuron. Actually, not necessarily it's -60, it can be from -60 to -70 millivolt. and minus 90 is the resting potential of a muscle cell, skeletal muscle cell. Smooth muscles have less negative potential. If potential moves upward to less negative values, this is called depolarization. And correspondingly, if potential moves to the more negative values, it's called hyperpolarization. Notice that it's different from mathematical comparison. For example, minus 40 is higher according to mathematics than minus 60. But in charges and potentials, minus 40 is less negative, less charged membrane. So we say it's a decreased potential, but better always to use not decrease or increase of potential, but better preferentially you should use less negative and more negative. And then it's easy to understand less is depolarization, more is hyperpolarization. So when line goes upward, this is depolarization. When it goes downward, according to the figure, it's hyperpolarization. Let's observe again how the sodium-potassium pump, or ATPase, operates. This is another way of presentation, but anyway, it's transfer, the same. Look, 3 sodium ions from inside are changed for 2 potassium ions from outside. It's easy to memorize which 3 ions and which 2 ions, because resulting negative charge appears after each cycle. So the cell should lose more positive ions than get back. ATPase portion splits ATP into ADP and phosphate, and this energy immediately changes the conformation so that 3 sodiums appears outside and 2 potassium ions are transferred inside. And again, we have one more negative charge in the cell. So it's electrogenic pump function. Thus, on the basis of all considered, we can state the main factors that determine the resting potential value. First, it's the difference of potassium concentration, because potassium goes out directly proportionally to the difference of concentrations. And the higher amount of potassium ions goes out of the cell, the more negative charge becomes inside. So the difference concentration is the factor that is directly proportional, determines directly proportionally the level of negativity inside, or resting membrane potential. Second factor is the permeability for potassium. Under normal conditions, it's a stable permeability, but it can be changed somehow a little bit more or less. And in this case, higher permeability provides more potassium loss from the cell and more negative or higher potential, hyperpolarization. Less permeability results in less loss of potassium and correspondingly less negative charge or depolarization. And these are passive factors. They don't need energy to maintain. So simply potassium goes out due to difference of concentration. And permeability does not require any active process. And just the third point, activity of the pump requires energy. And it's also directly proportional and determines the resting potential value, because when pump's activity is higher, it means more negative charge inside, because it's electrogenic potential. Naturally, pump activity does not increase higher than normal, but you may understand that as it depends on ATP availability, deficiency in ATP, such as can be created by hypoxia, results in less pump activity, and less activity results in less electrogenic action, less potential, or depolarization. So these are 3 main factors. And for the first 2, again, higher difference or higher permeability results in higher potential or hyperpolarization, and less results in less or depolarization. It's uneasy. And pump activity also. But for the first factor, difference in potassium concentration, sometimes it's necessary to start from consideration, not from difference immediately, but we consider how the changes of potassium concentration in the extracellular fluid may influence the resting potential. And it's important to estimate resting potential changes, because resting potential also determines the excitability of cells. So let's observe the influence of blood plasma potassium level. It's more variable as compared to intracellular concentration, and difference depends on both intracellular and extracellular concentration, and if potassium outside is changed. Let's begin first normal case. In blood plasma, as well as in the whole extracellular fluid, concentration is low, and therefore there is a normal difference of concentration, and therefore normal negativity inside and normal positive charge outside. So now let's consider that there is a hyperkalemia, high potassium level. We considered in the endocrine system that some hormones deficiency or excess may result in excessive potassium outside, like for example deficiency of aldosterone, and some other cases appear. So hyperkalemia means that here in the extracellular space concentration is higher, and if this concentration is higher, the difference becomes less, therefore less force to move for potassium outside. Less potassium goes outside, less negativity develops inside. Less negativity corresponds to depolarization. So in the figure, quoting from the normal level, this is normal potential, we move to depolarization, less negative. So the increase of blood plasma potassium concentration results in a resting potential decrease, which is referred to as depolarization. Let's consider the opposite case. If, as compared to norm, we have hypokalemia, decreased potassium concentration in the extracellular space. If it decreases, so difference becomes higher. Concentration difference for potassium increases, and it makes more potassium to go out of the cell, and therefore more uncompensated anions remain in the cell, and higher negativity develops at the inner side of membrane. More negative charge corresponds to hyperpolarization. From -60 it goes downward at the figure, becomes more negative as compared to normal. And more negative is hyperpolarization. So finally we can conclude that the decrease of blood plasma potassium results in a resting potential increase or hyperpolarization. So you see, it's rather logical and easy, just you should consider each time it in two steps. First, change of blood plasma concentration. Second step, how in this case the difference of concentration is changed. And then it's easy. Difference of concentration changes the same direction as the potential itself. If there is higher difference, like in the last example, there is higher potential or hyperpolarization. So if you consider it in two steps, you always easily can answer the question how blood plasma potassium level influences the potential of the excitable cells. So finally, conclusion. Blood plasma concentration increases, this results in gradient decrease, and so resting potential decreases too. Then plasma potassium concentration, when it's decreased, it increases the gradient, and so correspondingly for the resting potential, it increases and produces hyperpolarization. And the cells do not stay always in their resting state. And when time comes, from the level of resting potential with its charge minus, the cells make short, quick oscillations up to positive charges, which are action potentials. But this is entirely different story, and it is going to be considered in the second part. Thank you for your attention.